Every single day, an overwhelming sea of raw genetic data, we're talking billions of chemical signals, gets magically transformed into clear, life-saving clinical diagnoses. Welcome to The Explainer. Today, we're taking a journey right through the clinical next-generation sequencing, or NGS, pipeline to see exactly how that happens. So here is our roadmap for today. We are hitting 4 main stops. 1. Sequencing and raw data. 2. Alignment to the reference. 3. Finding variants. And 4. Clinical interpretation. Okay, let's dive right into this. You can really think of this entire pipeline like a massive data funnel. We start at the very top with billions of raw data points in what's called a FASTQ file. Then, through a series of computational steps, we filter all that noise down into a BAM file, then a VCF, and finally, those crucial actionable insights you see on a doctor's clinical report. It's honestly an incredible process of data reduction. All right, Section 1, Sequencing and Raw Data. Let's talk about actually generating those reads. When a sequencer, like an Illumina machine, processes physical DNA samples, it has a choice to make. It can do a single-end read, basically reading the DNA fragment from just one side. Now, that's cheaper for sure, but you get less information. Alternatively, it can do a paired-end read, reading the fragment from both ends. Paired-end sequencing is the most common method used in Illumina sequencing because, frankly, it provides a much higher degree of accuracy. And obviously, when a patient's health is on the line, that accuracy is exactly what we need. All those millions of reads get dumped directly into our first major file, the FASTQ file. And these things can easily be dozens of gigabytes in size. Think of it as the raw, completely unfiltered output straight from the machine. But what makes the FASTQ file so important isn't just the sequence data itself. It also holds a crucial grade for how accurate the machine actually thinks each base call is. And that accuracy grade? That is the Phred score, or Q score. Now, you might think a Q20, which means the sequencer is 99% sure it's right. Sounds pretty good. But in clinical NGS, 99% just isn't good enough. We're looking for the gold standard here, which is a Q30 score. A Q30 score means there was only a 1 in 1,000 chance of an error. That gives us 99.9% accuracy. In the clinic, we literally only want to rely on data that hits these incredibly high-quality metrics. So we've got our FASTQ file, millions of highly accurate, tiny pieces of DNA. But here's the catch. They are completely jumbled up. Where do they actually belong in the human body? Answering this question takes us from the raw data generation phase right into the puzzle assembly phase of our pipeline. Moving to Section 2, alignment to the reference genome, mapping those DNA reads. Using some really powerful software, think tools like BWA or Dragon, that raw FASTQ data gets mapped to a standard human reference genome like GRC37 or GRC38. We are essentially matching the patient's DNA to a master map of human DNA. Once that software does its heavy lifting, our raw data transforms into a BAM file, which stands for Binary Alignment Map. Because it's a binary file, it's highly compressed. But when paired with an index file, it allows analysts to instantly jump to specific regions of the genome and actually view those aligned reads manually using visualization tools like IGV. But how do we know we've aligned enough data to actually be confident in our results? Well, this introduces a critical concept: coverage, or depth. This simply means how many times a specific position in the DNA has been read by the sequencer. We also look at the breadth of coverage, which tells us exactly what percentage of our target region was successfully mapped. Like, did we actually cover 99.5% of the gene we're investigating? So the real question is, how much depth is actually required in the clinic? Well, if we're testing germline DNA, meaning the DNA you were born with, a depth of 100x is often totally sufficient. But cancer? Cancer is different. Somatic testing, which looks for mutations specifically inside a tumor, demands a massive 500x to 1,000x depth. We literally need to read that exact same spot of DNA up to 1,000 times just to ensure high clinical accuracy when searching for a tumor's specific mutations. Okay, so now that our patient's DNA is neatly aligned against the reference genome, how do we spot the typos? How do we find the exact errors that are causing the disease? This pushes us even further down our data funnel. Section 3, finding the genetic variants, spotting the mutations. Using specialized variant callers like GATK or Dragon, an absolutely massive data reduction happens right here. That giant BAM file, containing all the normal and abnormal DNA reads, gets completely stripped down. We essentially throw away all the normal data and we keep only the mutations. This creates the VCF, or Variant Call Format file. The VCF is hands down the most critical file in the entire pipeline because the final clinical report is going to be based entirely on the mutations listed right here. These variant callers are hunting for very specific typos. For instance, we might see an SNV where just a single nucleotide changes. We might find an indel, an insertion or deletion of sequences that can cause a really dangerous frame shift mutation. Then we have larger changes, like a CNV or copy number variation. Think of a HER2 amplification where entire copies of a gene multiply. And finally, structural variants or fusions, where two different genes break and fuse together, just like the BCR-ABL fusion commonly seen in blood cancers. Now, when we find these variants, we have to categorize where they came from. We very clearly contrast germline variants against somatic variants. A germline variant is present right from birth, it's inherited, and it is found in every single cell of the patient's body. A somatic variant, on the other hand, is an acquired mutation found only inside a tumor. And identifying those somatic variants is the primary target in cancer NGS. To figure out if a mutation is germline or somatic, we look at a really cool metric called VAF, variant allele frequency. This is just the percentage of total reads at a specific position that actually contain the mutation. If you inherit a germline mutation from one parent, you'd expect a VAF of right around 50%. But VAF is absolutely vital for cancer panels because somatic mutations might be present in only a tiny, tiny fraction of the tumor cells we biopsied. In clinical cancer sequencing, we're routinely hunting for a VAF of 5% or even lower. You're literally trying to find a needle in a biological haystack where 95% of the data is completely normal. So our pipeline has successfully found a mutation. It's sitting right there in the VCF file. But here is the most important question in medicine. Is it actually dangerous to the patient? I mean, this because DNA is different doesn't automatically mean it's deadly. Which brings us to Section 4, Clinical Interpretation and Reporting. This right here is where bioinformatics transitions into actual medicine. We start by decoding the variant through a process called annotation. Annotation software tells us exactly which gene is affected and what happens to the resulting protein. Did the mutation cause a missense change altering an amino acid, or was it a nonsense change, creating a premature stop codon that truncates the protein entirely? We then cross-reference that variant in massive public databases. For example, we check NOMAD. If a variant has a population frequency greater than 5% in healthy individuals, well, it's generally not considered the cause of a rare disease. We also check ClinVar, a global database where labs report whether a variant is actually connected to a specific illness. Armed with all that annotated data, clinical geneticists use guidelines from the ACMG, that's the American College of Medical Genetics and Genomics, to classify the variant into one of 5 distinct categories. Category 1 is pathogenic, meaning it 100% causes disease. Category 2 is likely pathogenic. On the complete opposite end, categories 4 and 5 are likely benign and benign, meaning they're totally harmless. But sitting right there in the middle is Category 3, VUS, or Variant of Uncertain Significance. This simply means we don't have enough data yet to know if it's dangerous or harmless. And honestly, that makes it the most confusing and frustrating result to see on a clinical report. But before we wrap up, we absolutely must remember one crucial thing. None of those ACMG clinical classifications matter if our foundational data is flawed. The entire clinical report rests completely on the quality control, or QC metrics, generated throughout our pipeline. Whether you're running Dragon or another clinical pipeline, verifying metrics like your Q30 scores, mean coverage, mapping rates, and variant allele frequencies is an absolute, non-negotiable requirement before a diagnosis is ever handed to a doctor. And that is the journey. Just take a second to think about the incredible scale of what we've just explored. We started with billions of raw chemical data points completely overwhelmed by noise. We aligned them, filtered them, and deeply analyzed them until we were left with just one single life-saving clinical insight. Mastering this massive data funnel isn't just a cool exercise in computer science. It's the absolute foundation that empowers the future of personalized medicine and targeted cancer therapies. And as AI continues to accelerate this massive data funnel, you have to wonder, what hidden cures might still be waiting to be discovered in your genetic code?